major phase 3 trial is bringing personalized cancer vaccination closer to clinical reality. Merck and Moderna report that Intismeran autogene, an individualized mRNA-based cancer vaccine given with pembrolizumab, significantly improved outcomes in patients with high-risk melanoma following surgical removal of their tumors. And the physiology behind it is remarkable. First, each patient's tumor is sequenced and compared with normal tissue to identify mutations unique to that cancer. Computational analysis then selects mutations capable of producing neoantigens, abnormal peptides that can distinguish tumor cells from healthy cells. Up to 34 of these personalized neoantigens are encoded into a synthetic mRNA vaccine manufactured specifically for that patient. After injection, the mRNA is translated into antigenic proteins. Dendritic cells process and present their peptides through MHC molecules, effectively teaching the patient's T-cells the molecular fingerprint of their own cancer. Pembrolizumab then targets PD-1, helping prevent the tumor from applying an inhibitory brake to those activated T-cells. Earlier 5-year data showed that the combination reduced the risk of recurrence or death by 49% and distant metastasis or death by 59% compared with pembrolizumab alone. Now, the much larger phase 3 INTERPATH-001 trial, enrolling more than 1,100 patients, has met both its recurrence-free and distant metastasis-free survival endpoints. Detailed phase 3 results are still forthcoming. This isn't CRISPR, and the patient's genome isn't being edited. Instead, tumor sequencing is being converted into a temporary set of molecular instructions that recruits the patient's own immune physiology against residual cancer. It represents an extraordinary evolution of precision medicine. Sequence the disease, identify what makes it unique, encode that information, and teach the immune system what to destroy.